we're going to talk about the exceptions to rules, all right? And so, you know, biology is not clean cut. You know, there's always some things that just don't act like everything else. And sometimes this happens in genetics. And the exceptions to this rule, the one that we're going to talk about first, is called incomplete dominance. Okay? And we talked about the blended theory and how before Mendel, you know, all the genes were thought to be just thrown into a big pot, and whatever came, the mixture of that would result in the offspring's traits, okay, what they would look like. And so before Mendel got into the act, that's sort of what we thought or think the mindset was, that how traits were passed on from generation to generation. Incomplete dominance is sort of like this mixing thing, okay? So if we have a monohybrid cross, when we start with a red, and plants show this a lot, okay? So snapdragon is a type of flower, right? If we start with a red snapdragon, we cross it with a white snapdragon, and they're both homozygous, okay? Then, in the F1 generation, we get heterozygous individuals, just like we would expect. However, their color is pink, all right? So it's a mixture. Carnations do the same thing. You get pink carnations this way as well, okay? White carnation, red carnation, pink carnation, all right? So when you take two pinks and you cross-fertilize them, you'll get one red, two pink, and one white, instead of three that are red. Okay? So this is called incomplete dominance. All right, when in heterozygous condition, the dominant allele does not always hide or mask the recessive allele, so there's sort of a mixture of them. Okay? Any questions about incomplete dominance? That one's pretty easy to think about. But the next one is called codominance. All right, and this one is going to be two different actual traits going on at the same time. So there's a little bit of complexity with it. All right, but you should star the definition of codominance. All right. And you should star ABO blood type, human blood type, okay? Because that's an example of that. And there are two multiple choice questions. One is that says a pair of non-identical alleles specify two phenotypes and both expressed in a heterozygous condition. That's the definition of codominance. And then there is the next question right after that is, give me an example of the one preceding, okay? So you need to know it's codominance, and then you need to give me the right example of that, and that's human blood type, ABO blood type, all right? We're not gonna look at the RH factor, positive or negative. We're just looking at whether you're AB, ABO, okay? All right, so in this example, you could either be O, little I, or something else. All right? Something else is up here. All right? So there's two, remember there's two, we're actually talking about two genes. So one gene is you're either O or not, just like eye color. We were talking about eye color, whether you had blue eyes, okay, blue eyes, or you have something else, okay, because there's more than one gene that we're talking about. So you're either O or something else, all right? The something else is dominant, okay? So, what can occur here is that we have a dominant I, big I, okay? And with that, we can have an A. Now, if we have two dominants together, we can have an A blood type. Or if we have a dominant A and an O, because the O is recessive, then it can be A blood type. Right? We can have the dominant being a B. If it's with a recessive or with an O, then it's going to be a B, but it also can be two dominant B's. Right? There is a situation where if you have a dominant A and a dominant B together, the A does not override the B and the B does not override the A. So they coexist, dominate. So in this case, you get AB blood, right? So that's where the codominance part kicks in, okay? Any questions about that?
So they can receive both A and B blood?
No, they had their own blood. They have AB
Yeah,
blood.
but if they needed blood.
They have to have AB blood,
Not A or B.
not A or B. Let me check on that. I'll check on it, but I'm pretty sure they have to have AB blood. But that's a point. That you have two alleles, and I think both have to be fulfilled. So you have to have, both of those being. I think you would now donating. Receiving, they'd have to have AB. I'm not sure about donating. But A is normally I think the most abundant, all right, positive, A positive. Others fall in, but AB is usually the rarer of blood types. Okay. Any questions about blood type? All right. And there are a couple other things that are exceptions to the rule. One is called pleiotropy. Pleiotropy is where you have a single gene, but it influences several phenotypes. Okay. So one gene causes sickle cell anemia, all right? But there are certain phenotypes that are part of sickle cell. The cells don't form correct, the red blood cells don't form correctly. There's some thinning of blood vessels. There's several phenotypes that occur because of one gene, all right? And they're all associated with the total sickle cell disease package, okay? Polygenic inheritance is where you have two or more genes influencing one phenotype. We're talking about eye color, right? Skin color, there's a lot of different, there's a lot of different phenotypes that are driven by more than one gene, okay? And when you have several genes influencing one phenotype, you have this gradation. You have a big sort of a, just a, they call it continuous variation, but it's just a whole mix of different shades or different types within the population, because the more genes, the more environmental factors, the more continuous the variation is going to be. The more types or variations you're going to have. All right, height is a good example. All right, there's all sorts of heights. All right, but because they're controlled by more than one gene, and there's other environmental factors involved, then you have differing types of people with their heights. Okay. Any questions with these? Okay. There is, now on your notes, there's some talk about Thomas Hunt Morgan and the location of genes on different chromosomes, right? We're just going to kind of skip over that. Just know that genes are found specifically on certain chromosomes, right? And those places are called loci, or locus is one. And the early work on this was done by this guy named Thomas Hunt Morgan, who is from Kentucky, but he did his work at Princeton. If you go to UK to their biology building, it's the Thomas Hunt Morgan biology building. Anyway, but on the sex chromosomes, the X and Y, there are genes found only on those sex chromosomes. And those particular chromosomes really lent themselves to this study of where these genes are located, how you can locate genes on certain chromosomes, because you only had these two, right? You either had two X's or an X and a Y. And so those genes that were associated with them, they could really intensify their research on. And so that's where they did a lot of their first studies on finding where these genes are located. And to that end, there is a gene on the Y chromosome, right? It's called the male determining gene, right? So during development, if that gene is present, then the reproductive tissue forms testes, all right? If that gene is not present, because there's an X there instead of a Y, then the reproductive tissue forms ovaries, all right? So that is an actual location, we know that's on the Y chromosome, that controls whether you're going to be male and female. And we talked about determining, you can, for a price, you can actually, more than likely get a baby of a certain sex. Because they can draw the Y chromosomes are smaller than the X chromosomes, so if you provide a sample, they take the top part of it, then more than likely they are going to have a Y chromosome in there, and then they put that into the female and hopefully get a male. If they want a female, they just dig down a little deeper in the specimen once they spin it down, because the heavier ones will fall to the bottom of the test tube when they spin it in a centrifuge at a high rate of speed, many G's. And so those that are found only on the Y chromosome, okay, or found only on the X chromosome, okay, are called sex-linked genes, okay? And so the Y-linked genes are found only on the Y chromosome. The X-linked genes are found only on the X sex chromosome. And the male determining gene is that one that we just talked about. Thomas Hunt Morgan did his research on fruit fly, eye color, and wings, whether they're fully developed or real wrinkly little kind of things. Anyway, he found that one of the eye colors was associated with the Y chromosome. All right. So the next little bit before we get to the review study guide deals with genetic disorders. All right. This really isn't in your chapter 9. It's something that I kind of I just decided to throw in because people are interested in it, right? And so there are things called genetic abnormalities. These are just versions of gene expressions that are in the population. They're just kind of rare, right? Infrequent. Genetic disorders are inherited conditions that result in mild or severe medical problems. They don't disappear from the population because there are mutations. Mutations are actual changes in the DNA sequence, right? So at a gene, if something is changed, then what happens is a new phenotype might arise, okay? So instead of fully rolling your tongue and not being able to roll your tongue, if a mutation occurred, then you can halfway roll your tongue, all right? I'm just making it up, but you know what I'm saying is, once a mutation occurs, different phenotypes can arise, okay? And then all within the population, you have heterozygous individuals, right? Those individuals carry that recessive or sometimes dominant allele with them, and they can pass that on to the next generation. That is why they used to ask for blood tests. I don't know if they still do when you get married. Okay. One, they want to make sure that your blood type, and especially the Rh factor, is the same. And two, they can do a karyotype if you're really interested. If you have something in your family history, some genetic disorder, and you don't have it, but you want to know if you can pass that on. So they will do a karyotype and see if you're a heterozygous for that, or if you're not carrying it. Okay. So a lot of that, especially if you're thinking about having children, they might ask you if you want to do a karyotype and just check and see, what the chances or risks that you would have for actually carrying a disease. And since we're heterozygous, we know that the genotypic ratio is 1 to 2 to 1, the 2 being the heterozygous condition, all right? So half of those individuals in the population could possibly be carrying a disorder, all right? So it is something to think about, all right? And so what we're going to do is we're going to talk about several different types of disorders, all right? This is the one that you need to remember. One of them, there's two, I'm sorry. This is the one of the two that you need to remember, all right? And what you need to remember is just an example. One of these three, okay? So it's called an autosomal recessive disorder, So these are found on one of the pairs of homologs, 1 through 22. Those are the autosomes. And then if you're homozygous recessive for that particular disorder, you get it, right? Albinism. Albinism is where you lack melanin in your skin, in your eyes. These people can survive. They just have to be very careful with the sun, with their skin, and with their eyes. But they have white hair, right? True albinos have pink eyes, right? So they just don't have that albinism. They're at high risk for skin and eye cancer. Sickle cell is a disease where the red blood cells don't completely form correctly, and so they sort of look like half moonish. They're not rounded, and so they don't carry the oxygen around in the hemoglobin like we talked about back in chapter 2 and in chapter 3. And so the people who have that disease are tired all the time. They don't get enough iron, they don't get enough oxygen, and therefore after several years their body just runs out because it's working so hard, right? You get organ damage. So most of the individuals who have sickle cell do die at an earlier age. All right, there's a lot of work being done. Those individuals who have darker skin and originally from tropical areas, okay, are at a higher risk factor for sickle cell than regular other people, okay? However, those that are heterozygous for that particular disease, they have their red blood cells form all right, but they are resistant to Plasmodium parasites, which cause malaria. Malaria is still the #1 killer of people in the world, okay? And their red blood, what it does is it gets into the bloodstream and it essentially eats red blood cells, okay? It goes into the cell and just explodes, okay? Those that are heterozygous, the carriers for sickle cell, they're resistant to the protozoa. Okay, it can't get into the cell. Therefore, they don't get sickle cell. Okay, and so this is an adaptation. Yes, it's bad if you are homozygous recessive for it, but if you're a heterozygous or homozygous dominant, you're resistant to malaria, which is more of a problem. Okay. The last one is called galactosemia, lactose intolerant. This is galactose intolerant. Galactose is another type of sugar, and you can't break it down. It can cause damage to organs if not treated, but it's highly treatable, okay, with diet and drugs. So just remember one of those 3, all right? The next one is autosomal dominant. So if you're homozygous dominant, you get the disease. Progeria is one where I think the movie was Jack, where the babies grow old exponentially. Okay, they have that disorder, they look like they're 90 when they're 8. Okay? They have very thin skin. Their eyes are sunk into their skull. Their teeth are more than likely gone. They have very scraggly hair. They have heart attacks before they reach maturity. There's about 500 cases of that, of kids living with this disorder in the world. So it's very, very rare. But it's caused by a mutation, all right? Something happened to the gene and it got mixed up, so it shows up. Huntington's disease, it's a deterioration of the nervous tissue, all right? It is those individuals usually don't live too much past maturity. It's a progressive disorder, so it cuts lifespan way down. Achondroplasia is one that results in little people. Okay. Most of the time, if it's homozygous dominant, their individuals are stillbirth. Okay, so they don't even become they don't get born. Heterozygous individuals don't have the proper cartilage formation, so they're abnormally shortened arms and legs. Most of the time, everything else is fine. And just about the stillbirth thing, your body, the woman's body, in this case, is extremely just brilliant thing, okay? Because here the developing fetus is developing in the body. The body knows if something's wrong or not, right? And a lot of times, if you don't have something right, the baby just doesn't develop, right? Because there's something wrong. And so those tend to be stillbirth, right? We went through a stillbirth after our 2 children were born, and it's just the way the body knows there's something wrong, and it just shuts down development. Okay, not all the time, but it's an extraordinary thing.
Do they miscarry
Huh?
sometimes? Does the body miscarry at some time?
They miscarry. The dwarf, especially if you're homozygous dominant for achondroplasia, that more times than not they're stillbirth. All right, there are some disorders that are connected to the X sex chromosome. Okay, and one of these is recessive. These two are recessive. One of them is hemophilia. It's a blood clotting